Welcome to week 8. So this week we are focusing on gram-negative diplococci. The nice thing is it's going to be a short little lecture, so not as many organisms to learn this week as there has been in other weeks. So the world of gram-negative diplococci for what we need to know is pretty small, which is kind of refreshing as we're getting toward the end of the first half of microbiology. So general characteristics, as I said, gram stain, of course, gram-negative diplococci, meaning they occur in pairs. The pairs are said to have looked like kidney bean shaped or coffee bean shaped. You'd have to have a really good microscope, of course, a really nice zoom focus, like to get in there and kind of really see that shape. In this image, you're not going to see the kidney bean or coffee bean shape like you would under a really good microscope. But that is said to be the shape of gram-negative diplococci occurring in those pairs. So where the black arrows are pointing, you can see, especially this center arrow here, you can see the two little gram-negative cocci in pair there. So with the world of gram-negatives, so this is kind of our first foray into gram-negatives. We've been dealing with all gram-positives up until this point. The first test you run when you get a gram-negative gram stain is an oxidase test. So that'll be the first time we're delving into the world of oxidase. So anytime we have a gram-negative cocci, gram-negative rod, we're running oxidase next. So gram-negative diplococci tend to be oxidase positive. So that's bolded there. And they also prefer an increased CO2 environment. So they love a capnophilic environment. So in the case of our student lab, if we're ever thinking we're looking for one of these, we would put these in a candle jar or a CO2 incubator to help them thrive even more. So there's another look at the gram stain. You can definitely see these ones a bit better than the other one was a little zoomed out. So definitely pairs. I mean, there's this one looks like it's in a foursome there, but otherwise you can see them all in little pairs here together and very much that pink coloring for gram negative. All right, so when we talk gram-negative diplococci, we're talking about Neisseриas and one Moraxella catarrhalis mainly. And that's the genuses that we are looking at. So starting with Neisseria, one of the big bad boys out of the Neisseria bunch is Neisseria meningitidis. The nice thing is the name tells you what it kind of tends to cause, which is meningitis. So meningitis has been heavily linked with Neisseria meningitidis. It can colonize our mucous membranes, definitely spread person-to-person respiratory droplets, that kind of thing. This does have a vaccine for it. So there is a vaccine already created to help prevent Neisseria meningitidis. It is done for children, but heavily wanted before you even get to like close quarter living like with teenagers. So before this vaccine, there was a lot of people in dorm rooms that could end up getting Neisseria meningitidis with those teenagers living in close quarters, sharing all those lovely germs. So they thankfully have come up with a vaccine preventing it, so that really has reduced down the number of times that we hear somebody in college getting sick with meningitis. And it's a very ill, the illness is very serious. It's prognosis is not always great depending on how quickly they can get seen, depending on if they have any other diseases or disorders underlying, how their immune system functions. But it is a very serious illness when they become sick. I mean, meningitis is always serious, but this Neisseria meningitidis, it does its job. The other main Neisseria pathogen is Neisseria gonorrhea. Again, the name tells you what it causes, gonorrhea. So, of course, not part of our normal flora. Neisseria meningitidis is also not part of our normal flora. You'll notice it said colonizing there. It doesn't exist as part of us. So neither one of these should be part of our normal flora. There are other Neisseria species that are part of our normal flora that can be found like in our oropharyngeal normal flora, but that is not meningitis or gonorrhea. That's other Neisseria species. So this is, of course, spread through sexual contact. I think pretty much everybody has tended to hear of gonorrhea. It is considered an STD. Actually, the new term is SID. So STD stands for sexually transmitted disease. The new term is STI. Sorry, I said that wrong. Sexually transmitted infection is like kind of the newer terminology now. So it is an STI. The D comes in, the disease comes in when that infection causes harm to that person, and then it becomes the disease. So that's kind of the difference, sexually transmitted infection. And then once that infection takes place and causes harm to any part of them, then it becomes the full-blown STD. So gonorrhea, of course, causes infections in genitals, rectum, and throat, since it is sexually passed. So there is a look at Neisseria gonorrhea. You'll notice this time it is inside the segmented neutrophils, or what you can call polymorphonuclear neutrophils, PMNs. You can hear them named either way, segmented neutrophils or PMNs. So it is seen as intracellular bacteria inside there, and they're all, of course, still in the little pairs. I can see them nicely here in this one right here where I have my cursor. Pairs right there, gram-negative diplococci. If you were to get a swab in from a genital rectal and it's showing this, that right there is already presumptive alone for knowing it's going to most likely be Neisseria gonorrhea because of the source it's coming from, the rectal, genital area. And then seeing gram-negative diplococci, again, we don't have that many pathogens that are gram-negative diplococci. So you're kind of already putting together the presumptive findings of it. Of course, we would still do our due diligence and test all the way for it. In the labs nowadays for gonorrhea, we are testing gonorrhea along with chlamydia. It's pretty much always ran together. Chlamydia, it's what we call like a GC test. Chlamydia gonorrhea test, but done by PCR. So that way the result can come back really rapidly. We're not culturing this anymore really in the lab. We're running it by PCR to look for it. And that way the result comes pretty quickly. But of course, it can be cultured. Same with Neisseria meningitidis can be cultured, but a lot of times we've gone to, running cerebrospinal fluids. Anytime we suspect meningitis, we can run a cerebrospinal fluid through, the DNA molecular process of if they have one of those MALDI-TOFs, which are a molecular analyzer or something else. BioFire is another one that does molecular testing currently, and there's a bunch of others. And on that CSF panel, they're looking for all the main causes of meningitis. So Neisseria meningitidis would be on there. Streptococcus agalactiae might be on there. Listeria monocytogenes might be on there. Staph aureus, all the big pathogens that we've already started to learn, plus others, that we haven't fully covered yet. So just keep that in mind that we're not always culturing everything, but we still have to learn the culture characteristics and the testing characteristics of these organisms. And then the third pathogen in this group is Moraxella catarrhalis. Now, all the other Moraxella species are gram-negative rods. Moraxella catarrhalis is the gram-negative diplococci, which is why it's in this unit. Everything else for Moraxella, like there's Moraxella lacunata, and there's some others, they are gram-negative rods. They might pop up time to time causing an infection, but we don't really focus on them. They're not as heavily seen as this one would be. So this is normal flora of our upper respiratory tract. That is also where it likes to cause infections. So Moraxella catarrhalis is a huge cause of upper respiratory infections, which is what URI is. So think otitis media, which is ear infections, sinusitis, so sinus infections, and it can go down into the lungs into pneumonia. This one has unique characteristic on the agar plates that they're large, medium to large gray colonies, but they have what we call a hockey puck consistency. So if you think about a hockey puck on ice, it slides across the ice. And that's kind of what Moraxella catarrhalis is known to do, is when you take your loop, you can almost slide the colony on the agar a little bit like that hockey puck. It can move across that agar if you're just pushing it just right, and it is Moraxella catarrhalis. So that is a unique feature that hardly anything else has. The Neisserias, I didn't really mention their colony characteristics just because they're gray. They're not that exciting to, on agar, but all these are more fastidious organisms, and Neisserias especially are very fastidious organisms. So with that, they are not going to grow on blood agar. They want to grow on chocolate because chocolate is our agar that will grow fastidious organisms. We also have a couple extra specialized media to grow, especially for the Neisserias. So we have modified Theriamartin and Martin Lewis agar. Both of these are used to look and screen for Neisseria. So in these agars, they are both chocolate-based agars that have a bunch of antibiotics in them. And the antibiotics ensure that nothing else really is growing except that Neisseria. So in the Theriamartin, it has colistin, which inhibits gram-negatives from growing. So it'll inhibit all the other gram-neg rods from growing. Vancomycin, which inhibits your gram-positives from growing. Trimethoprim inhibits a specific organism called Proteus. Proteus is just a gram-neg rod that really swarms and takes over a plate. So we can use trimethoprim to kind of decrease that action. And then nystatin will inhibit any yeast from growing. So that way, by the end of it, you're pretty much just left with gram-negative cocces left to hopefully grow if they are there. So that way only Neisseria is coming through, and all the other kind of normal flora and anything else that might have been in that genital area or wherever you're looking can thrive. And so they're wiped out, and just the Neisseria can thrive and really be seen. Same with Martin Lewis. It has very much the similar antibiotics. The difference is it has an increased amount of vancomycin, and then it uses mycin. Sometimes it's called anisomycin to inhibit the yeast instead of nystatin. So I do need you to memorize these antibiotics on each of these augurs and what they inhibit. That is important for testing, question-wise, exam-wise. So knowing what's in each of these, what they inhibit, and of course that they are special augurs, they're specific augurs for the growth of Neisseria. There is a third augur. Sometimes you hear it. It's called the NYC augur. Think of like New York City, NYC augur, that also can be used to grow Neisseria. I don't test on the NYC one, but I do use the saying. And again, we're all college age if you're listening to this, listening to my lecture. So I will tell you my saying that I use to help remember what grows Neisseria. And it's not a fun saying, but I say the two Martins, so Thayer Martin and Martin Lewis, go to New York City and get gonorrhea. So that's how I remember that these three augurs are used to grow Neisseria. So if that helps you, use it. All right, as far as testing wise goes, like I said, we always oxidase test first as soon as we know we have a gram-negative. In this case, it would be oxidase positive for these organisms. The big thing with Neisseria is the moment that we're suspecting a Neisseria, because one, it's facetious and only grow in chocolate, and then also take into account your source, if it's coming from a genital rectal throat or if it's coming from a CSF or meningitis. You know, it could be also the Marxella coming from like an upper respiratory type thing. But if it's facetious, only certain, and you're getting a gram-negative diplococci, oxidase positive, then you want to look at carbohydrate testing, sugar utilization. The Neisserias love to use sugars, but only certain ones depending on the organism. So Neisseria gonorrhea will use up glucose, but not maltose, lactose, or sucrose. All of those would be negative. So I always put the G's together, gonorrhea, glucose, positive. Neisseria meningitidis will utilize glucose and maltose, but not lactose or sucrose. I know I don't have sucrose on here, but you get it. So I always put the M's together. It goes out to the M's, both the G plus the M now for meningitidis. There is another Neisseria called Neisseria lactamica, very low virulence, doesn't cause hardly anything for disease-wise. But if you were to test it, it's named lactamica because it can use the lactose sugar. So in this case, it's all three get utilized. Sucrose would be negative. Now there is another Neisseria that would use all the way out to sucrose. So you can see how that sugar utilization really separates between the Neisseria organisms. So there are little carbohydrate test kits that can be run. I think in I don't understand. I think in your student lab, you might not have enough time to run them because they take several hours of incubation, that kind of thing. But at some point you might be able to get a chance to see them. It'll just depend on the setup and what you can do in your student lab. So it is important to remember those sugar test differentiate between those Neisserias. Now, if you did have a Marxella catarrhalis growing in this case, it would not use any of these. It would be negative for all the sugars. And then you can do a different test called a DNase test. So that is an enzyme that Marxella catarrhalis does have. And that enzyme breaks down DNA into smaller pieces. So there is a DNase augur that you can use and streak out your suspected organism on. And so on that augur, after the DNA is broken down, it will no longer bind to the methyl green in the augur and it'll make a halo effect. So like letter A here would be a positive DNase. Letter C would be negative. B would also be positive. So C here is negative, A would be positive, B would be positive, because you see the haloing effect. So there's methyl green in this augur, which is what makes it that blue-green color. And so once that DNase enzyme has enacted and broken down the DNA in it, it can't bind to that green color and produces a halo effect. So that's how it pops up positive. So that's one way that you could test for DNase. Again, Marxella catarrhalis will be very much positive for DNase. The Neisserias will be negative. So that's another way you can separate between these gram-negative diplococces. So again, oxidase testing, you'll do this with a dropper in your lab. You can put it onto a disc like this. You can also do it right onto the auger plate. Like a purple color is positive for oxidase. So if they have that enzyme, it'll show up as a purple color. there is for the most part, oxidase should always be purple, depending on, I don't know if there's another type of oxidase test run that's a different one, but we always know it as purple coloring that for a positive. There is a flow charting system, of course, anytime you suspect Neisseria or Moraxella catarrhalis. I know this is really fuzzy looking, so I apologize. I couldn't get it to get clear. But again, you're always thinking Neisseria or Moraxella catarrhalis when it's a gram-negative diplococci. And then over here, again, it's showing your main pathogens is Neisseria gonorrhea, Neisseria meningitidis. And then there is a bunch of others, like I said, that are just normal oral floras a lot of times. That is typically where they're carried, but they're listed down there. So, but again, they are very much oxidase positive. You can perform sugar utilization the minute you know it's gram-negative diplococci and DNase to separate out the Moraxella catarrhalis as well. So not a lot for testing wise to remember here. Thankfully, there's only 3 main organisms that you have to remember, and they all, 2 of them are named for what they tend to cause for infection. Moraxella catarrhalis is the one that just causes, in general, upper respiratory and some pneumonia. So a little bit lighter of a week for us, which is always appreciated. That is it for week 8. I hope you guys have a great week.